Okay, so hello everyone. I'm very happy to introduce to you Alina Selega today. She's a postdoc research fellow at the Lundvall Research Institute in Toronto, and she works with Kieran Campbell in machine learning for genomics. Her research interest lies in Bayesian optimization for strategies of heuristic objectives, mainly for biomedical applications, and therefore her title is going to be or talk about multi-objective Bayesian optimization with heuristic objectives for biomedical and molecular data analysis workflows. So without further ado, the stage is yours, and I'm very excited to have you here today. Thank you.
Thank you so much for the introduction. Yeah, you already kind of said everything I was going to say here. You read my title. Thank you. It's a bit of a long title, but hopefully at the end of the talk, we will all understand what that means. But yeah, my name is Alina. Nice to meet you all. Thank you so much for giving us this opportunity to tell you about our recent work. It's recently got published in Transactions on Machine Learning Research journal this March. And yeah, happy to join you virtually from Toronto, but let's begin. So as a little overview for my talk, I'm going to start by setting the scene and talking a little bit about the context of AutoML and AutoML related approaches in the domain of bioinformatics, and kind of touch upon some successes and some outstanding challenges. And I'm going to use this discussion to motivate our work because we really try to address a specific kind of typical scenario that arises when working with the bioinformatics workflow. I'm going to go through some definitions, and then I'm going to introduce our method, which is called Manatee. I'm going to illustrate it on some synthetic toy data and also on the real data set that corresponds to a real bioinformatics workflow. I'm going to close with some conclusions. Okay, so I'm going to start with a kind of a success story. So this is a type of a problem where Bayesian optimization, a related AutoML approach, has been applied successfully within bioinformatics. And this problem is protein design. So the aim of protein design is to find a protein sequence that will have some desirable function that we care about. And so proteins are just chains of amino acids kind of assembled together, and they can vary quite dramatically in length. They can be hundreds of amino acids long or thousands. And there are 20 amino acids that participate in the functional genomic code, and they can be assembled in any order. So this already gives you a little bit of an idea of a challenge for this problem. So if we did consider the whole space of all possible protein sequences, only a tiny minority of that space is actually represents functional proteins that would exist in a cell or maybe perform some function. So when let's say we actually found some sequence that we think is a promising one, so then to actually find out if the sequence maps to our function in a way that we want, we need to do a cost expensive screening step. So typically, given a protein sequence, it will get synthesized in experimental conditions in a lab. We will make this protein and then do some sort of perform the screening where we can score the sequence on our function that we want to measure. So for this reason, Bayesian optimization has been applied to this problem within this kind of cartoon workflow here. I'm going to assume that you can see my mouse. I think that typically is how it works. So we start with a parent molecule, which is just some sort of protein sequence. And this is typically kind of selected intelligently. So maybe we already have most of the sequence down because this is a protein that belongs to a certain family and we already know roughly what the sequence is. But the idea is that sometimes making small changes, like substituting some amino acids for others, can actually dramatically improve the function that we want to measure. For example, if it's like some sort of biophysical properties of a protein, it could make a molecule more stable, or if it's maybe binding affinity, it can bind its target better. So this typically is performed with this mutagenesis step, where we take our parent and we make these little changes to it when we just swap some amino acids for others, creating this variant pool of protein candidates that we then screen via this experimental step, and we generate these sequence function pairs, where each sequence that we are considering has now a mapping to the function. And this feeds into the sequence function model, where on the x-axis we have the protein sequences, and on the y-axis is the measure of the function, with higher being better. And then we can use Bayesian optimization kind of in a standard approach to give us sequences that we should try and do apply the mutagenesis step again. And then we can kind of continue doing this iterative process for as long as we want. And so Bayesian optimization has been used to optimize protein thermostability. And this was actually a paper from 2013. I think that was the first time that Bayesian optimization has been applied to this problem of protein design. And since then, it has been really, yeah, quite an effective tool. And as recently as this year, another paper also used Bayesian optimization with some other bells and whistles to optimize binding affinity of an antibody for its target. So these approaches were able to leverage Bayesian optimization because they have access to this black box oracle that they can target, that they can query and then score the candidates that we want to evaluate on this function that has been optimized. However, this is typically not the case in most of the bioinformatic analyses context. And this is because bioinformatic analyses are, in their nature, almost always unsupervised and very exploratory. So biomedical data, which is the input to bioinformatic analyses, is typically high dimensional. And in this talk today, I'm going to specifically focus on gene expression data. That's also where I mostly work within. So gene expression, when we measure that within a cell, what we do is we quantify the cell's phenotype, or kind of roughly speaking, we measure what the cell is doing, how it is behaving in its surroundings. And so when we measure gene expression in many cells, a common analysis for this type of data aims to group cells by their expression profiles. So we end up with this kind of matrix, data matrix, where on the rows we have genes, and we can have something like 20,000 genes. That is a kind of typical number if we subset to protein genes. And then on the columns we have cells, and we can have thousands of hundreds of thousands of cells, or maybe even more, depending on what kind of data set we're working with. And we want to take this data and cluster it and identify groupings of cells based on their expression profiles. And typically these groups correspond to cell types or maybe cell states. And by cell state, I just mean subtypes within a cell type. So the cells are still of the same cell type, but they act in some different ways. And that kind of corresponds to their state. So there are a lot of different cell types in any organism. So we have T cells, for example. T cells are a part of your immune system, and they are actually able to kill infected cells. So they can help us if there's any kind of infection that we're dealing with. We've got epithelial cells. These are the cells that line surfaces within your body, everywhere, inside and outside. And there's a lot of other cell types that all perform different functions. So why do I actually care about it? Why is it important to know what kind of cell types we have in a sample? Why do we want to know how they act? Well, it turns out that actually knowing what kind of cell types we have and how they behave and kind of act and interact with each other is a very strong predictor for a lot of important clinical features. It can be a predictor of treatment response. It can be a predictor of disease subtype or disease severity. So in general, this is a very important problem in bioinformatics. Okay, so when we are kind of doing this annotation, I guess, so when we take our gene expression data and we want to find these kind of groupings of cells within it, we don't have any ground truth labels that we can compare with, right? We have to do this annotation in this unsupervised way. And even more than that, any existing labels that people deposit and are out there in public access, when people publish a new data set, they can annotate it and also post labels. These can also be a little bit unreliable, just to make the matters harder. Why is that? Well, annotations are typically done by a manual annotation. So it's an expert that looks at these cells and says, these are cell type A, this is cell type B. And this could be a little subjective because an expert might be using specific information when they're doing that. And another expert might annotate a little bit differently. But also, this annotation is always performed on the output of some bioinformatic workflow that has been run with a certain hyperparameter setting that the computational biologist that ran it has chosen. And so just to have a look at what that looks like. So a typical workflow, bioinformatic workflow involves many steps, and all of these steps have parameters that are associated with them. So between our input data and the result, whatever we want to get from our workflow, we can broadly categorize these steps into data processing steps and then analysis steps. And analysis is actually what our workflow is supposed to be doing. And here I'm kind of collectively denoting all of the parameters across all of these steps as this one variable theta. So let's consider an example. So a clustering workflow that we already kind of talked a little bit about before. So our input data might be some sort of single cell expression data where we measure expression of single cells, and then we want to cluster, right? So we want to get cell clusters as a result. So then when we're doing data processing, we might choose to filter our data for quality control, like filter out poor quality cells. We might choose our favorite normalization method to normalize our data. select our favorite clustering algorithm, set all of the parameters for all of these different steps, and voila, we have our cell clusters. Here's the problem. So different parameter values, especially when combining across many different steps, can actually lead to very different results. So I might set these parameters to different values and then end up with cell clusters that really look quite different. And also not just in terms of like number of clusters, but also in the membership of cells that get assigned to different groups. And this is kind of intuitively somewhat clear, right? When we have a data set that is two different data sets that are acquired in different experimental conditions, we might, for instance, do different kind of filtering for each one, right? Maybe we need to do more stringent filtering for one and less stringent for another to like get rid of the correct amount of noise and then end up with cell clusters that are gonna maybe better separate cell types that are closely related in terms of their expression profile. So in a nutshell, there is no one right way to set the parameters of a given workflow that will work equally well for all data sets that you have. So what do we do with this? Okay, so computational biologists that typically run these workflows in practice, including myself, what we honestly do is we try some values, and typically only a few, because some of these workflows can take a long time to run. So we set some parameters, we compute the results, and then we evaluate the success of the results. So in our clustering example, the true kind of objective that we might want to benchmark against is, did I find biologically meaningful cell types? This is a very complex question that is very hard to answer, and it's impossible to answer in a single way, right? So for this reason, instead of that, practitioners might turn to easier to define objectives. For example, do the clusters that I found, cells within those clusters, do they express known proteins? Because, for example, from like the literature, we know that certain cell types will express one protein that we know about, and then other cell types will not express that protein. So that is one objective that we can turn to, but also many others, as you can probably imagine, are possible. And here we're kind of getting towards perhaps the biggest challenge of applying AutoML and AutoML related approaches to bioinformatics, specifically for hyperparameter optimization of bioinformatic workflows. And it's the difficulty with defining objectives. It's very hard to define a single kind of true objective, but even defining many is kind of a little hard. So first of all, you have a lack of ground truth that we've covered, right? And then, yeah, you have many, many possible measures that you could use to quantify the success of your workflow. And we talk about them as these heuristic measures, and I'm going to illustrate what I mean by that. So in this table here, I just have some recent studies that all, as part of their work, benchmark a single-cell RNA-seq clustering workflow, right? They just have a workflow and they want to see whether the clusters that they find are good or not. And so this is the different objectives that they turn to. So it's quite typical to turn to expert labels. I know I have told you already that they can be a bit unreliable, but unreliable labels are better than no labels. So often people use some sort of concordance measure with expert labels. So this is adjusted RAND index, have been used by these two studies. But then different studies can use a lot of different things. So for instance, this top one used also silhouette width, so like a cluster purity metric. Then the second study used like a handcrafted heuristic that was centered around ability to detect rare cell types, because this is something that they really cared about in their study. And then the final one also evaluated the robustness to model misspecification. So for instance, if you told your if you told your AutoML pipeline that there are 3 cell types that you want to kind of identify, but then actually one of them is not in there, how does your model deal with that? Or if you didn't mention a cell type that actually your data does have, so how does the model deal with that? So this is just to show that our community really turns to these heuristic measures that are subject to user preference in lieu of any kind of better ground truth measures, because we just simply do not have those. And just to, again, make the matters harder, we also have to deal with unexpected experimental failures in the data generating process. So what I mean by that is kind of illustrated in this cartoon here. So here on the x-axis, I just have some sort of parameter of my workflow, theta, doesn't matter which one, that I'm trying to optimize. And then I have, when I'm evaluating my clusters that I got, right, I decided to turn to these known proteins that some cell types should express, right? So I have 3 here, okay? And then each objective measures kind of some degree of separation of cells that express that protein. And if this degree of separation is high, that means that the cells that express this one protein are separated from others, And that's good, right? Because this is kind of what biologically is meaningful and maps onto my domain knowledge. So I might end up with the situation, and this actually happens frequently all the time, where measuring a certain protein failed in your experiment for experimental reasons, okay? So you might end up with observations that look a little bit like this one here on the left, this box on the left. So when I fit my favorite model to it, I'll end up with a really flat mean and really, really high variance. And then I don't really know which parameter setting is good to choose to optimize this particular objective. So in this particular case, I might be better off just throwing this F1 objective altogether and then focusing on my other two. So these are the two proteins that I successfully did measure in my experiment. And then, I should choose a parameter of like whatever that is, 0.25 for my theta, because this is where my kind of heuristic objectives are maximized. So this is like a kind of a cartoon motivation for these unexpected failures. But my point is that not only do we have a lot of possible heuristic measures of workflow success, some of them might actually not be very useful in the overall optimization procedure. Okay, so how do we actually, what do we do with this? There are a handful of approaches that are AutoML related that try to tackle this hyperparameter optimization problem, or at least parts of the pipeline optimization. And how they deal with that is that you just have to do your absolute best to create really good objectives for your specific problem. So an example for that is a method from 2021 called AutoGeneS. And this method aims to deconvolve bulk RNA sequencing data into contributions from different cell types. So what is bulk RNA-seq? That's just gene expression measured across many cells. So it's like averaged across all the cells that you had in the sample from all the different cell types. And it is an important problem to try and deconvolve it into cell type contributions. So normally how this works, you have to specify which cell types you want. So this is here illustrated by these colors. So you have some sort of reference data that quantifies expression profile for those cell types. And then let's ignore this box for a second. Typically what happens then, this gets distilled into the signature matrix where it's normally of lower dimensionality. And you hopefully will only keep genes that really separate your cell types, so that you can really, when you're trying to do this kind of regression analysis, you can say this cell is from this cell type, this cell is from this cell type, because the signature matrix hopefully includes only very well discriminating features for genes. So yeah, once you have the signature matrix, you take your bulk data and you regress it on this matrix and then get your cell type proportions. So you had this much of this cell type, this much of this cell type, etc. So selecting these well discriminating genes is a complicated problem, and people do a lot of different things for it. But what AutoGeneS did, they decided to use multi-objective optimization to do this feature selection step. And so how they did this, they constructed two objectives that, broadly speaking, measured cell type separation and then optimized those to perform this feature selection step. So specifically, they wanted to select genes such that the correlation between cell types is minimized and the distance between cell types is maximized. And a little motivating cartoon is here on the right. So the top left box is the most desirable case, that's the one that they want. So on the axis, we have the expression of a gene, and we have a pair of genes here. So this would be the discriminating genes that we are selecting possibly, right? And different circles represent cells and the expression, and we've measured the expression of that gene in that cell. And then the colors correspond to cell types, right? So you can see that this is a good case, right? So if we had this case, that would mean that gene 1 and gene 2 are good discriminators between the cell types, because each cell type expresses one of the genes highly and does not express the other. So that's great. They're lowly correlated, and the distance in this expression space between the cell types is high. And also most cells in one cell type express that gene in at similar levels, right? So they're all kind of clustered together. So it's low variance, and that's also a good situation to have. But then, yeah, this is all of the other cases where things can go wrong. You can have highly correlated things, or very close in the expression space cell types, or you can have really high variance where within the same cell type, some cells express the gene really highly, but some really lowly, and then you can have this overlap. So that would not be such a good discriminating feature. So that's kind of the motivation for it. And yeah, that's kind of the crux of what they did. AutoGeneS were able to leverage multi-objective optimization because they had access, well, they were able to construct really well-defined, very informative objectives for this one specific given problem of feature selection in the problem of Baltic convolution. Now you can actually notice here that they talk about very low variance as a useful, like desirable characteristic, right? But they actually decided not to include it as a third objective. So they only focus on these two objectives in their optimization procedure. And they instead just filtered out genes with high variance before that. And this again just kind of demonstrates that even in this case, when they were able to come up with these well-defined informative objectives, we could still include more objectives, but we don't really know which ones are going to give you better performance. And one could venture a guess that this gave them better performance, but who knows? Okay, and finally, this kind of I'm ready to describe the motivation for our work. So our work is really motivated by the case, and we aim to address the case where we have many potential objectives that can measure workflow success, but we do not know upfront how well they measure our success, right? So yeah, in the case of our clustering example, our heuristic objectives could be like, do the clusters express non-proteins? Are the clusters well separated? Or any other way that you might assess whether your clusterings are good or not. And so all of these heuristic objectives are designed, or they're supposed to approximate the true meta-objective, did I find biologically meaningful cell types? But the meta-objective is kind of impossible to quantify, to be honest. But not all of them, not all of these heuristic objectives may be useful. And so when I say useful here, what I mean is not all of them are guaranteed to align with the meta-objective with respect to their maxima. And we want those that do align, so that when we optimize our heuristics, heuristic objectives, we will also recover the maxima of the true meta-objective. So to try and address this case with this uncertainty about the usefulness of the heuristic objectives, we designed a method that automatically infers which objectives are useful, and I'm using the kind of definition for useful from the previous slide. And then we use this inference about which objectives are useful to guide optimization towards the maxima of those useful objectives. So we do that by letting the user express preferences over the functional form of the objectives. And I will talk about this more, but we kind of postulate this by this concept of desirable behaviors that we kind of, this is one of our contributions, is introducing this concept. So these desirable behaviors are expressing the user preferences, and they are designed to describe practitioners' expectations about what a useful objective should look like. We compute these behaviors from the posterior of the surrogate function, and then our method, which is a multi-objective Bayesian optimization method, uses this to automatically concentrate on the region of the solution space that maximizes the objectives that we have inferred as useful. Okay. So I'm sure this is very familiar because this is the AutoML seminar series, but just to be complete about it, I'm just going to super quickly fly through some basics for multi-objective Bayesian optimization so that all the definitions are clear. So in a typical Bayesian optimization situation, we might have some observations shown here in this little crosses, and we want to know where to sample our function next, right? Because it's expensive, etc. We want to find the best next place that we should sample our function. So the true function is shown as kind of lime green, and then we fit our favorite model to our observations, and we end up with the mean and uncertainty information. And then typically what is done is we use some sort of cheap to compute acquisition function that will take this information from our model and kind of combine it together such that the higher values of the acquisition function correspond to the desirable regions of our space where we could sample next. So optimizing the acquisition function gives us the next place where we should sample. We acquire our next observation and continue. So when we're dealing with a multi-objective case, we have multiple objectives to reckon with instead of just one. And one of the approaches to kind of combine them together is the scalarization function approach. And I denote it as S lambda. And these scalarization functions are typically parameterized by weights lambda. And then they kind of combine objective values or acquisitions of the multiple objectives into a single scalar for a given value of x. So this is just some kind of weighted expression that combines values from your different objectives, and then you can optimize that instead. Okay, so when we're also dealing with multiple objectives, it is kind of expected, and often is the case, that they can be conflicting or they could have maxima in different places. So we typically talk about the Pareto friend, and that's the set of all points at which no objective can be improved without degrading the other. And theoretically speaking, they all are kind of equal optimality. So we could return any point from the Pareto front, right? Theoretically speaking, that's all good. However, in applied problems, it is actually often desirable to only focus on a subset of the Pareto front, some region of it. And here's an example for it. In drug design, one of our objectives might be toxicity of a drug, and we might not be willing to go beyond a certain toxicity. So like if a drug is too toxic, that would just kill our patients, and we can't do that. So then kind of these constraints on the what objective values we will deem acceptable will also translate into the constraints on the Pareto front, and then we would have to only look at some part of it. And this is the idea that we build on in our method. So we build on the approach by Perea et al. From 2019. And that's the approach that uses random scalarizations that return a subset of the Pareto front. So how this is done, they parameterize their scalarization weights, so the weights lambda that the scalarization function uses. They impose this distribution P of lambda that is actually specified by the user, and they provide some ideas of how to specify that. And so this P of lambda determines the desired region of the Pareto front that we're going to get solutions from. Here's a nice handy cartoon from their paper. So we have two objectives on the both axes, and the solid black line corresponds to the Pareto front. And then the dashed line kind of shows you this imposed distribution P of lambda. And so when we optimize, when we perform optimization under this construction with these imposed distribution of the scalarization weights, we end up with samples shown here in blue that are returned where the P of lambda kind of tells them to be, and therefore we only focus on the part of the Pareto front and not the whole Pareto front. So this is kind of how we can constrain our optimization with imposing this distribution over scalarization weights. So we do kind of, we follow the similar approach, but instead of just asking the user to specify P of lambda, we determine the region of the Pareto front using these objective behaviors that I've mentioned before. So the idea is that we want to return solutions from the region of the Pareto front where the useful objectives are maximized. Now, how do we do that? We infer which objectives are useful using objective behaviors. And so the idea is that a useful objective will have a desirable behavior. And then what happens is that we upweight useful objectives in our optimization procedure. And I'll show that in more detail. And in our framework, behaviors are user specified, but we do propose some examples in our work. And this is the 3 examples that we propose. So the first behavior is explainability, and that captures how much an objective covaries with the parameter that we're optimizing. And we favor those objectives that have lower observation noise versus high observation noise. This kind of the cartoon shows it here. Second one is inter-objective agreement, and that basically favors objectives that agree with others. And the idea is that, we're hoping that all of these objectives should really kind of measure similar things, or at least be maximized in similar regions. But if there's a few that agree, but maybe we can be a bit more confident in them that they weren't noisy or didn't fail in the experimental conditions. And then the third and final one is maximum not at boundary. And so this determines whether the maximum of the objective is contained within the user specified parameter range. So the user specified parameter range is another input to our method because we need to know the bounds for the optimization for the parameter in question. And I guess the idea here is that if we end up with this situation here on the right, that the objective function kind of decreases or increases with the parameter value, then either that maybe the user specified the parameter range wrong and we should have continued more to the left to find where the actual maximum is, or maybe this objective is just entirely unbounded in this parameter, and then it's not really going to be useful for us in the optimization procedure. All right, so now with these kind of, this is the examples for the desirable behaviors that we propose. Again, these are not the only ones, and they might not apply everywhere, but this is some sensible examples that we feel can work for a good number of workflows. And then now in more detail, this is how we actually, this is what we do in our method. So we first define a set of behaviors, and this is, I showed you the examples just before, and I denote them as this bold B, and the subscript K corresponds to the objective K. Then we condition the distribution over scalarization weights on the objective behaviors, right? Because we want to focus on the region of the Pareto front conditioned on the behaviors that we've computed from these posteriors of our objectives. So this is our new conditional distribution over scalarization weights, and the calligraphic B is just a set of all of the behaviors of all of the objectives. Then we do something different, and we define objective weights, lambda k, so this is for each objective k, we define them to be binary. And lambda k equals 1 indicates whether an objective is useful, and 0 indicates that it's not. Then we also define conditional distributions over behaviors given a useful or not useful objective. So this is P of bk given lambda k equals 1 for a useful objective, or 0 for a non-useful objective. And this describes what do we expect this behavior that we've defined to look like for a useful objective and a non-useful objective. And then we use these conditional distributions together with the prior to compute the posterior probability for an objective being useful. So this P of lambda k equals 1 given its behaviors. And then finally, we use a linear scalarization function as lambda with this expression down here, but it's just a weighted sum. And then when we use this linear scalarization, we actually end up having these above mentioned posterior probabilities for an objective being useful act as objective weights. And so if we have a posterior probability for a given objective that it's useful to be quite high, it ends up with a high weight. So we upweight it in our optimization procedure. And if it's a lower probability, then it's downweighted. Okay, these two steps are user specified. So the definition of the behaviors and their also definition of the conditional distributions over behaviors is user specified, but we propose some examples in our paper. Okay, so this is the overview of our method, and it's called MANATY, and it stands for Multi-Objective Bayesian Optimization with Heuristic Objectives. And I know this is a very contrived acronym, but we get a cute acronym, so it's worth it. So it is a multi-objective Bayesian optimization method. So it kind of follows the typical multi-objective Bayesian optimization workflow. We start in step 1 when we acquire some data, and we have k heuristic objectives, and not all of them may be useful. We then fit a multi-output Gaussian process model to it. In step 3, we infer objective behaviors, which are the 3 behaviors that I've introduced, and they are computed from the posterior form of each objective. Then we compute these posterior probabilities for an objective being useful that act as objective weights in our algorithm. And then we use these weights to update our overall optimized expression. So here, this is an expected scalarized acquisition function, and it's shown in green. And so when we optimize that, that tells us where we should sample next, and we acquire the next point and continue for the desired number of steps. Okay, so the first evaluation is just based on toy data, just to give an intuition of how this works. And the idea here is that we simulated 3 objectives that are simulating the useful objective case, and then 2 that are kind of not useful. So the first 3 signs are kind of the useful objective. So they're all sign forms, except this one is a ReLU sign and also has a larger noise than these 2. But these are kind of the useful objective, the meta objective of the sign, and these are the useful heuristics. And then we have just random noise that is slightly shifted. So it has this kind of slightly like values closer to 0 are slightly higher than those that are closer to 1 in the parameter space. And then the final one is just a linear function x equals y. So I'm showing here in blue and yellow samples from these objectives, and the yellow are the samples that lie in the Pareto front for these 5 functions. And then in red, I'm showing the acquisitions made by MANATY over 30 steps, including also the initial training set of 5 points. So we can see that MANATY acquires points that land on the maximum of the signs, because these are the objectives that it has inferred are useful. And so in the bottom row, I'm showing the posterior probability for an objective being useful conditioned on its behaviors. So with the 3 signs, these are quite high probabilities. And then for the 2 non-useful objectives, the probabilities of being useful are lower. And so that's why we're able to maximize these 3 useful objectives. That is reflected in the acquisitions. Okay, and now we can move on to the real workflow. So this is a workflow for the analysis of imaging mass cytometry data. And imaging mass cytometry is a technology that profiles the expression of roughly 40 proteins at single cell level while preserving spatial information. And the raw data that comes out of the technology is routinely normalized with this inverse hyperbolic sine function, where we take the raw data and we divide it by this normalization parameter called cofactor C, and then it gets put through the strict function. And so there is currently, it's still a new technology, and yeah, there is no systematic way to set C, and it is definitely kind of with the same considerations plausible that different values of C could lead to better dissection of clusters and separation of similar cell types for different data sets. So as one of the contributions of our paper, we construct a set of example objectives for this problem, and we apply our method to optimize the cofactor C in a clustering workflow for an existing IMC dataset that was published in 2020. And so, yeah, this type of IMC data also gets typically clustered to recover cell types. Okay, so the objectives that we construct for this clustering workflow, we do this by using known protein pairs that we know to be highly expressed in one cell type and lowly in all others. And we define our objective as the correlation between mean cluster expression of the proteins in a pair. So let's imagine, so here in this cartoon, I have on x-axis expression of protein 1 and on y expression of protein 2. That's my pair of proteins that are supposed to be co-expressed in a given cell type. So if we have successfully separated cells that express these two proteins in their own clusters, then if I plotted the mean cluster expression, they would land here. This like pink cluster would contain all these cells and purple cluster would contain all these cells, and they would land in this part of the space with a high expression of both proteins. And then all other cells that don't express it would be down here, right? So when we computed the correlation for the scatter plot, we would have a high correlation, and that would indicate a good clustering. And then if we have cellular populations that are mixed, so the cells that express or do not express these proteins are all over the place, mixed across the clusters, we would end up with low correlation and a bad clustering. And in this example, we have 7 such pairs and 7 objectives. And so, yeah, the optimization problem is we want to optimize the cofactor C, and then we make acquisitions as follows. We set C to a certain value, we normalize our data, we cluster our normalized data, and then we compute these correlations for our objective functions on the acquired clustering. Okay, so we also turn to expert annotations like everybody does. So this data set does have expert annotations. And using those, we constructed 2 meta objectives, which basically measure the concordance between the expert annotations and the cluster labels that we get when we cluster our data in this unsupervised way. So here we use adjusted rand index and normalized mutual information. And on x-axis, you see different values of the cofactor. And on the y-axis is the concordance between the clustering that we obtained for that given cofactor value and the expert annotations. And you can see that the lower values of the cofactor, so we considered a range between 1 and 100, lower values around 1, 2, something like that, maximize both meta objectives. Okay, and what we found when we applied our method is that the parameters that were selected by our method led to clusterings that agree with expert annotations. So much like I was showing before with the toy example, in the top row, I'm showing all of our different objectives. So this is our 7 co-expression-based objectives for different protein pairs. And again, the blue and the yellow are the samples from the functions, and the yellow are the Pareto optimal samples. And the red show the acquisitions by our method. And so you can see that our method acquires points mostly that kind of overlapping in the lower end of the range, agreeing with where the meta objectives are maximized. And in the bottom row, we also see the probabilities, posterior probabilities for an objective being useful. So for example, if we look at this objective here, it's actually maximized in the opposite range, right? So the values are higher for really large values of the cofactor for this particular protein pair. And so the probability for it being useful has been inferred to be lower. And these are plotted as a function of the acquisition step. Okay, and yeah, so for the quantitative evaluation of our performance, we also constructed 3 regret-based metrics. So we decided not to use the hypervolume improvement as our main evaluation because in our setup, some of the objectives are not useful. So it doesn't really map exactly to our scenario. So we just, again, still used our meta objectives, and I denote them as HX. So it's either the adjusted random index or the normalized mutual information with expert annotations. And we denote the overall max as Y star. That's just the overall max of the given meta objective. And so we have these 3 different regrets that are all defined in terms of the overall max Y star and also the value of the meta objective for a given cofactor value, for a given acquisition. So for a certain cofactor value XT, we would have done the whole workflow, compute the clustering, and then compute the concordance with the expert annotation. And so this is the table up here when we compare with some baselines and some state-of-the-art methods that I'm happy to talk about after the talk. And The top rows are two of our method formulations that all follow the same kind of workflow, but slightly different formulation of the optimized expression. And you can see that we outperform other methods on this example. And also at the bottom, the plot here shows the kind of cumulative versions of these regrets, where we just plot them as function of the acquisition step and only consider the acquisitions made to a certain step. And our curves are shown in orange and blue. And you can see that for cumulative regret, we actually acquire lower regret value. And for full and base, the curves, the blue and orange curves decrease a little faster than others. So kind of as another cumulative metric. And yeah, just as an aside, meta objectives are only available in benchmarking exercises. Here we are benchmarking. If we were ever doing this to a new data set, we wouldn't have any expert annotations and would not be able to do any of this analysis. Okay, so sorry, maybe 2 minutes over time, but yeah, so, okay, good. In summary, our method is designed for scenarios where many objectives are possible and kind of heuristic objectives because they are subject to user preference, but we do not know upfront which ones are going to end up being useful in terms of aligning with the meta objective that we really do care about in terms of their maxima. In our framework, a practitioner defines desirable behaviors and what these would look like for useful objectives. And we propose some examples for what behaviors could be sensible choices. A practitioner defines these only once and does not participate during acquisition in our framework. So this is all just done once at the beginning, and then the rest is automatic. And our method infers which objectives are useful based on behaviors that we learn from the posterior fit. And then the optimization is guided towards maxima of those useful objectives. And also kind of this feeds into our main contributions. So we introduce the concept of behaviors, that they can be computed from the posterior fit. We suggest examples of such behaviors. We in fact infer objective usefulness via conditioning the distribution of scalarization weights on these behaviors. And then we also construct some objectives and apply our method to real biomedical workflows. I've only showed you results from one, but we also consider a second case study in our paper. And the final note is just to say we obviously talk a lot about heuristics in here and the like subjectivity, and our method is not magic. It does not get rid of subjectivity. The subjectivity just all gets pushed up kind of a level, and it all lands in the definitions for behaviors and the conditional distributions over these behaviors. So instead of answering a question, which objectives should I use to benchmark my workflow, the question now becomes, what would the behavior of a good objective be? And this is kind of what we try to address with our method. Yeah, with this, I really like to thank you for your attention and very happy to answer your questions. Thank you.